Right. Hello, everyone. My name is Hani Fismail. I'm speaking on behalf of UCL-TB. Welcome to our BCG Centenary Symposium. Just waiting for the participant numbers to come up. I think we had just over 300 people registering and about 120 of you have just joined so far. So I guess first thing to say is happy birthday, BCG. 100 years ago yesterday, the first dose of BCG was given to an infant whose mother had just died of tuberculosis at L'Hôpital de la Charité in Paris. And it still remains the only licensed vaccine against TB and is the most widely used vaccination in the world. But TB still remains one of the leading causes of death from infectious disease. So UCL-TB and the Africa Health Research Institute are delighted that we can co-host this BCG Centenary Symposium. And we're very grateful to our three fantastic speakers, Mark Hatherill from the University of Cape Town, Trish Darrah from the NIH, Alex Schmidt from Gates MRI for joining us in this. And through their talks, we're hoping to celebrate BCG, reflect on what BCG vaccination has achieved over the last 100 years, explore what we're still learning from the host response to BCG, and look at the pipeline of vaccinations, which we hope will facilitate control of this disease. So followed by these talks, there'll be a panel discussion with our three speakers, which will be led by Willem Hanekom, who's director at the Africa Health Research Institute. And he'll be using your audience questions as a starting point for this discussion with the help of Lele Rangaka, who will also be curating these. I'm sure you're all by now familiar with this Zoom webinar platform. So if you have a question for the speaker, you need to put that into the question and answer box rather than the chat box. And then you'll be able to vote on these questions and they'll move up and down the list. And then we can identify from those the questions that will be put to the panel. If any of the questions are not the speakers, we'll be able to respond to them directly in a written way in the question and answer tab. So we hope that you're going to be able to stay with us for the next 90 minutes and enjoy the symposium. I'm just going to hand over now to my co-chair from AHRI, who's Indira Govinda, who's going to introduce our first speaker. So thank you very much and enjoy.
Thank you, Hanif, and welcome to everyone who's joined us today. I have the pleasure of introducing our first speaker, Professor Mark Hatherill, who's the director and leader of the clinical research team of the South African Tuberculosis Vaccine Initiative, known as SATVI, at the University of Cape Town. Prof. Hatherill is a University of Cape Town medical graduate, and he's trained as a pediatrician and a critical care subspecialist. Since 2005, SATVI has conducted 28 clinical trials of 9 novel TB vaccine candidates involving almost 6,000 participants, in addition to large epidemiological and immunological cohort studies. Prof. Hatherill played a lead role in efficacy trials that tested the candidate vaccine MVA-85A for prevention of TB disease in infants, BCG revaccination for prevention of Mycobacterium tuberculosis infection in adolescents, and the candidate vaccine M72AS01E for prevention of TB disease in adults. He is the co-principal investigator of the Regional Prospective Observational Research in Tuberculosis South Africa Consortium, and his current work focuses on the design and implementation of clinical trials of novel TB vaccines and biomarker-targeted TB prevention. So I'd like to hand you over to Prof. Hatherill, who will do his presentation. Thank you.
Thank you very much, Indira, for that kind introduction. So I must say it's a great pleasure to speak at this BCG Centenary Symposium. To kick off, I've got a relatively short time to cover 100 years of BCG. So I'm going to try and highlight some of the most important developments through the last 100 years, and also to try and give you a flavor, a contemporaneous perspective of how these developments were viewed at the time. So Let's begin at the beginning then, and go back to before BCG, so to around 1990. This is the TB prevention toolkit. This is a set of TB control guidelines for the prevention of TB in children. And you'll see that some of the concepts are fairly familiar: social distancing, cough hygiene, improved ventilation. Some of the other concepts are possibly more related to parenting than TB control. So do not pick your nose with your fingers or eat with dirty hands. But the point being that the TB prevention toolkit in the early years of the 1900s was very limited. So it was in this setting that two scientists, doctors Albert Calmette, a physician, and Camille Guérin, a veterinarian, who were working in the Pasteur Institute in Lille, France. And they developed a live attenuated bovine TB strain using a strain of M. bovis from a cow with mastitis. They cultured this strain, and because their cultures were becoming contaminated, they tried using bile as a natural detergent, and they unexpectedly discovered that the organism lost virulence over multiple in vitro passages. And this organism that would become BCG was tested for safety in a number of animal models over the subsequent years. It was only in 1921 that Calmette declared that he was satisfied that BCG would not again regain virulence as a human pathogen, but still they were reluctant to test BCG in humans. Until Dr. Weilhaller, who was a physician in Paris, came to them with this case of a newborn whose mother had died, and he would be forced to live with his grandmother, who had active tuberculosis. And as they say, he was doomed to infection with tubercle bacilli. Bearing in mind that in the early 1900s, the mortality rate for infants who became TB infected in the early months of life was somewhere between 50 and 70%. And so they made the decision to give BCG, which was given as an oral preparation on the 3rd, 5th, and 7th day of life. There were no obvious ill effects in this case series of n equals 1. And the child remained healthy and did not develop TB, despite, as they comment, being brought up in a bacilliferous milieu. And it was these kinds of uncontrolled experiments that were practiced over the subsequent years. And it was only in 1931 that the numbers had accrued to such an extent that Calmette could report on cohorts of infants who had been vaccinated or not vaccinated with BCG. So by that stage, they had accrued more than 8,000 young children who had been exposed shortly after birth in the household. And he reported all-cause mortality in the BCG vaccinated group as 4.6%, as compared to between 16 and 25% in the unvaccinated group. There was still some skepticism from their contemporaries, and primarily relating to the potential risk of regaining virulence. But it was only in the subsequent 20 or even 30 years that randomized controlled trials that we would recognize provided solid evidence for the efficacy of BCG in infants. And here's one. So you're looking at data from an elegant trial performed by Sol Rosenthal in Chicago in the 1940s and 50s. You're seeing in the figure controls, those are TB cases. The circles are TB cases occurring in the control group above the horizontal line, and cases amongst the BCG vaccinated beneath. And it's very striking that up until the age of 2, there are in fact no TB cases occurring in TB vaccinated infants. So already convincing evidence in infants of protection against TB disease. We also should remember that the cases occurring at that time in these trials were in the pre-chemotherapy era. So unfortunately, what was being observed during these trials was the natural history and the natural outcome of childhood tuberculosis, such that many of these cases progressed to post-mortem or calcification, and the endpoints included miliary TB, TB meningitis, and TB deaths. But because of that, we're able to answer the question of whether BCG protects against these severe forms of TB in young children. And from this systematic review, we have an estimate of 64% efficacy against death from TB and 64% efficacy against tuberculous meningitis in children, as well as 78% efficacy against disseminated TB. So convincing evidence that BCG is beneficial in young children. Does BCG protect children against all types of TB, including intrathoracic or pulmonary TB? Here we have evidence from 4 large trials conducted between the late 1940s through until the early 1960s. And in these four trials, the pooled estimate of efficacy was 74% against all forms of tuberculosis. What about adults? So we've seen convincing evidence of protection in children, and at the same time in the early 1930s, 1940s, we have some evidence from natural experiments, such as this one from Norway. So this report describes a group of student nurses, almost 1,500 student nurses, who came to work at a hospital in Ullevål between the late 1920s and the early 1940s. Slightly more than half of them were tuberculin skin test negative on arrival at the hospital, and this group demonstrated 100% tuberculin skin test conversion after 3 years of working at Ullevål Hospital, which gives you some sense of the TB burden and the transmission setting in that hospital setting. What we also see is that amongst these TST negative nurses, who all become what we would call then recent converters within a fairly short time, there were those who were BCG naive, had an astronomical TB case incidence of slightly more than 14 per 100,000, and a TB mortality of 1.4, so this is per 100 person years, of 1.4 per 100 person years. What then of those nurses who received BCG? Well, we can see here that of those tuberculin skin test negative nurses who received BCG, the TB case incidence and TB mortality was 6 to 7-fold lower. So very similar to what had been seen in children in similar uncontrolled experiments. But remember, there were also half of the group of nurses who were TST positive and presumably TB infected on arrival at Ullevål Hospital. And these nurses did not receive BCG. And in comparison with their contemporaries who were BCG naive, skin test negative, and who then recently converted, those who had previously been converted also had a much reduced rate of both TB and TB mortality. So this raises the question then, whether BCG vaccination of TST positive or older people can improve on the protection afforded by latent TB infection. So let's dig into that a little bit further. So some of the trials to help us answer this question include what's known as the Chingleput trial. So this was a massive trial conducted in South India for the prevention of culture-confirmed TB disease. Of the more than 260,000 participants, the majority were older than 15 years of age, although there were some children. and more than half were tuberculin skin test positive. And in this massive trial, there was no evidence of protection against microbiologically confirmed TB disease in this study population in this setting. So how do we reconcile then this compelling evidence of protection in infants versus this lack of protection in the Chingleput study population? So systematic reviews will be helpful in addressing then the results from a number of studies. And here we have a particularly elegant systematic review from Poonam Mangtani and colleagues who showed very convincingly that the protection offered by BCG vaccination against TB disease is significantly greater for vaccination of infants and tuberculin skin test negative children, as outlined at the top of the red box, as compared to adults, whether tuberculin skin test positive or negative. And it's not known whether this effect is due to blocking of BCG replication due to prior mycobacterial immunity or masking of a protective effect by prior MTB immunity. So you'll also have noticed from the previous figure, perhaps, that there seemed to be an effect by latitude. And here we're also able to answer this question in this systematic review. And this group of investigators showed that although BCG protection was absent or low, very low, in trials conducted at latitudes less than 20 degrees, as well as those conducted between latitudes of between 20 and 40 degrees, as compared to those trials conducted above 40 degrees latitude. It's not known whether this effect is due to some factor of the population, of the setting, of exposure, or is this due to trial design bias or trial quality? But these investigators were able to answer this question to a certain extent, because indeed, when one adjusts for age at BCG vaccination, TST status, and diagnostic detection bias, then the effect of latitude is attenuated and becomes no longer statistically significant. So let's pause then there and reflect on the impact of BCG on TB mortality and morbidity. So, for example, if we compare mid-19th century England and Wales, we have TB mortality estimated at 600 per 100,000 children. And even in the 1950s, infant TB mortality was approximately 50%, with up to 20% of infected infants developing tuberculous meningitis. Fast forward to the 1980s, 1990s in South Africa, which is only 10 to 20 years after the rollout of BCG to infants. And we already see that death due to TB and tuberculous meningitis amounts to less than 1% of childhood TB cases. And in the modern era, in the 2000s, in the clinical trial setting, at least, there are very few deaths due to TB, and tuberculous meningitis is a rare event. And it's largely for this reason that the BCG Atlas looks as it does. So in the countries in orange, BCG is given universally to infants through most of the countries of South America, Africa, and Asia. However, if we contrast this map of BCG use with the map showing estimated TB incidence rates, we see the highest TB incidence rates across the globe are in fact in these same regions. So it becomes apparent that infant BCG vaccination has had little impact on the TB epidemic amongst adults, which is important because adults or adult type TB is the driver of TB transmission. And this brings us then to, in my view at least, what is the primary limitation of BCG. And that is not that BCG is not effective against tuberculosis, because it is. It is the fact that the protection offered by BCG is not durable. And this systematic review, or at least we have two systematic reviews here, it becomes apparent that the protection offered by BCG rarely lasts for more than 10 years. There are some notable exceptions, one trial in Brazil, one trial in Alaska, where protection lasted through and is measurable into the 40s. But by and large, vaccination of infants with BCG does not offer protection beyond adolescence. We've discussed several of the limitations of BCG. Are there any other benefits? And certainly it has been or has become accepted over the last several years that there are off-target or non-specific heterologous benefits of BCG vaccination through the mechanism of trained immunity, which is the induction of non-specific memory mediated by epigenetic and metabolic rewiring. We have a few reports demonstrating that such BCG-induced innate immunity provides protection against viral infections in mice and in vivo evidence of reduction in experimental viral titers in humans. We have some data from human studies to suggest that BCG is associated with protection against respiratory tract infections, but the data are not easily comparable across study designs and populations. So the evidence for a consistent clinically meaningful benefit for prevention of viral infections is somewhat limited. A slightly more compelling evidence that BCG vaccination of infants is associated with a reduction in all-cause childhood mortality, and particularly in that age group prior to receipt of the other killed or inactivated vaccines. And that effect is observable in both TB-exposed and TB-unexposed children, as compared to the children who did not receive neonatal BCG. And this is one of those curious facts that, in fact, we are learning again, because it was pointed out by Calmette in 1931 that tuberculosis mortality becomes nearly zero and general mortality is 4 times less than it is in non-vaccinated children in areas where BCG is rolled out. That effect does not, however, appear to relate to BCG vaccination of adults or last long term. There are other intriguing, potentially beneficial effects of BCG. To give one example, I'm showing at the top on that figure on the right-hand side, an 8-year randomized controlled trial of the use of BCG, which is shown in red, for treatment of type 1 diabetes. And you'll see there that there was no or very little effect for 3 years, but after 3 years, there was a significant reduction almost to normal levels of HbA1c, which is a marker of glucose control. There have been some reports of a beneficial effect of BCG vaccination against all-cause wheeze and childhood asthma, but the data is not conclusive. But what is conclusive is that BCG installation for a non-invasive bladder cancer is best practice management, and the data are robust to suggest that intravesical BCG is in fact more effective than chemotherapy for this use. So take-home messages then. We've heard that BCG vaccination of infants offers protection and conclusively offers protection against childhood TB. We've seen that vaccination of older or particularly skin test positive people offers little or no protection. And notably, we've seen that BCG-induced protection is rarely durable into adolescence. On the other hand, BCG does seem to have intriguing non-specific or off-target effects, but the magnitude and consistency of the clinical benefits arising from those effects remains somewhat uncertain. And all of these considerations have implications for novel TB vaccine candidates that seek to replace or add to the BCG immunization schedule. So I'd like to acknowledge conversations with many collaborators, including some on this call. And I'll leave you with the one thought that I remain torn as to whether we should celebrate the centenary of BCG in terms of the millions of lives that have been saved, or lament the fact that our TB vaccine technology in 2021 is 100 years old, and we are driving the Model T Ford of TB vaccines when we should be driving a Ferrari. Maybe it's a little of both, but I will stop there, and thank you.
Thanks, Mark. That was absolutely fantastic. Perfect timing as well, with my alarm going off in the background. Remarkable to go through all of that history in 20 minutes and some great thoughts at the end. So if you've got any questions for Mark, please do put them in the question and answer tab, and we'll be able to pick them up in the discussion. So I'm just going to introduce our next speaker now, who is Dr. Trish Darrah. Tricia received a PhD in microbiology and immunology from Temple University School of Medicine in 2001. And since then, she's worked in the cellular immunology section of the NIH Vaccine Research Center, studying how various vaccine platforms shape the magnitude, quality, breadth, and tissue distribution of CD4 and CD8 T cell responses after immunization. Tricia currently leads the VRC-TB unit, and she is investigating the immunogenicity and efficacy of new vaccines against TB in the preclinical non-human primate model. And she's going to talk to us about this in a talk entitled Lessons Learned from Protective Immunity from IV BCG in Non-Human Primates. Many thanks. Over to you, Tricia.
Thank you. Let's see. Okay, good afternoon, and thank you for the opportunity for talking to you today. The work I'll show today is a collaboration between the NIH Vaccine Research Center and Joanne Flynn's lab at the University of Pittsburgh. So today I'll be discussing 3 non-human primate studies, first reviewing our published and initial findings showing that IV BCG was protective in non-human primates against TB, then a dose de-escalation study in NHPs to define correlates of protection, and finally a T cell depletion study to try to understand mechanisms of protection. So several years ago now, we began asking the question of whether the route and dose of BCG would influence immunity and protection against TB in non-human primates. We delivered a high dose of BCG, either intradermally, aerosolly, or intravenously, followed by a low dose MTB challenge 6 months later, and found that intravenous BCG really conferred dramatic protection. Here you see that animals that received intravenous BCG had significantly lower lung CFUs at the time of necropsy compared to these animals that received the standard human dose of ID BCG, whereas animals that received the same high dose of ID or aerosol BCG did not protect. In fact, 6 out of 10 of these animals had sterile protection or undetectable CFUs at the time of necropsy. This protection was associated with high frequencies of antigen-specific CD4 and CD8 T cells, detectable in the bronchoalveolar lavage, or BAL, shown here in red, as well as recruiting a high proportion of tissue-resident T cells in the lung, which we were able to determine using this intravascular CD45 staining technique, which discriminates between tissue-resident and blood-resident cells. Here in these flow cytometry plots, you can see a representative animal that received ID BCG or aerosol BCG. And in comparison to those animals, an animal that received IV BCG has this really high number and frequency of tissue-resident T cells that we can detect in each of the lung lobes analyzed. So IV BCG is significantly more protective than the same dose given by ID or aerosol, and elicited the highest number of TB-specific T cells and tissue-resident cells in the lung, as well as having higher antibodies in the serum and lung, which I didn't show you. Importantly, IV BCG provides a benchmark vaccine to study immune correlates and mechanisms of protection in non-human primates. So to try to determine or define immune correlates of protection, we set up the following study in which 35 animals were immunized with half-log increasing doses of intravenous BCG between 5 times 10 to the 4th and 3 times 10 to the 7th. And this highest dose was just slightly lower than the dose used in the published work that was 5 times 10 to the 7th CFU, but I'll include those animals in my analysis for comparison. So the idea behind this study was to use different IV BCG doses to elicit a range of immune responses to which we could correlate differential outcomes in protection. And in fact, we were able to generate a range of immune responses using this dose escalation study. Here I'm showing you that the number of antigen-specific CD4 T cells or CD8 T cells in the bowel or antibody titers in the bowel all positively correlated with as the BCG dose increased. However, these correlations were not perfect. For example, in this medium BCG dose, you can see one animal has a very high response that's higher than some animals that received even a higher BCG dose, whereas another animal in that same group has a much lower response than some animals that received even lower BCG doses. This slide shows the outcome for that experiment. On the right, you can see PET CT images arranged from top to bottom with increasing BCG vaccine dose. Again, the orange areas here are areas of TB disease. On the left, I'm showing you total mycobacterial CFUs at necropsy. So unvaccinated animals have about 10 to the 6th CFUs at necropsy. In comparison, we saw a high degree of protection across all doses of IV BCG. In fact, about 50% of these animals had no detectable TB disease at necropsy, including even one animal that received this really low dose of 4 times 10 to the 5 BCG. So taken together with our historical data, that's about 23 out of 44 animals that have sterile protection or undetectable protection after IV BCG, and an additional 4 animals that had very low disease at necropsy. So using this data, we can perform logistic regression analysis and show that in general, the level of protection increases along with the IV BCG dose. And we can also try to estimate a 50% protective dose of IV BCG, which comes in as just under 10 to the 6th CFU. Thinking about this data, irrespective of dose, we saw that there was an inverse correlation between total CFU and necropsy and the number of antigen-specific CD4s or CD8s in the bowel, as well as with the antibody titers in the bowel. However, and out of the 3 immune parameters, you can see that the CD4 responses had the highest R-square value. However, if you look in each one of these groups, you can see exceptions to this correlation curve. For example, here's an animal with a high response, but a low degree of protection, where here are 2 animals that have low responses, but have sterile protection against TB, indicating perhaps, not surprisingly, that protection against TB may be multifactorial. We've recently analyzed the blood responses in these animals, and that's data shown on this slide. So although we did see a hierarchy of antigen-specific blood CD4 T cells shown across the decreasing BCG doses labeled here from A to F over time, we failed to see the same inverse correlation between total CFUs and the antigen-specific CD4 T cells in the blood, like we had seen in the bowel in the previous slide, at least by the single immune parameter that I'm showing here, which is cytokine production of gamma-2 TNF or IL-17 production. So with the continuing ongoing correlates analysis will be to take a really multidimensional systems approach where we're going to incorporate all immune parameters measured both in the blood and the bowel, as well as whole blood transcriptional analysis. However, I'd remind you that correlates that we find may not be mechanistic. We'd really like to know what immune parameters are mediating protection against TB after IV BCG. There's at least 3 mechanisms of protection after IV BCG, including T cells, antibodies, and trained innate immunity. However, our group has really focused on whether or not T cells are required to mediate protection after IV BCG. So T cell depletion in rhesus macaques can be accomplished using depleting antibodies such as anti-CD4 and anti-CD8 alpha. Depleting CD4s is fairly straightforward. This antibody targets single positive CD4 T cells as well as double positive CD4, CD8 positive T cells. However, the CD8 alpha antibody is a little bit more tricky. That antibody is going to target these double positive cells as well as any cell that expresses CD8 alpha, which includes all classical CD8 T cells that express CD8 alpha beta, in addition to these non-classical CD8 alpha alpha expressing cells, such as NK cells, gamma deltas, mates, and iNKTs. So this depletion study involved 39 animals, all that received IV BCG, as well as 3 unvaccinated infection controls. 5 months after vaccination, these animals were split into 3 groups that received either control IgG or saline, or anti-CD4 or anti-CD8 alpha depleting antibody. Antibody infusions began 5 months after vaccination and 1 month prior to challenge, and continued every 2 weeks throughout the challenge phase. And then in the next few slides, I'll show you the antigen-specific responses in the bowel after vaccination, the extent of depletion after antibody infusions began, and then the protective outcome. So as we've shown in our published work, but can now show with many more animals, IV BCG induces a 10 to 50-fold increase in the total number of T cells that can be harvested from the bowel over the 2 to 12 weeks that we monitored this compartment. That influx results in a dramatic shift in the proportion of macrophages, which predominate the bowel prior to immunization, and T cells, which predominate the bowel after vaccination, essentially resulting in an inversion of this effector target ratio. And that inversion remains imprinted for at least 3 months after vaccination. Looking at only the T cells in the bowel, we see that there's a rapid and transient increase in the proportions of V gamma 9 gamma delta cells, as well as mate cells, that occurs 2 weeks after immunization. However, by 4 and 8 weeks after vaccination, these proportions have returned to pre-vaccination levels. And at this 12-week time point, you can see what you're left with is a large proportion of CD4 T cells compared to CD8 T cells compared to those pre-vaccination proportions. Moving to the antigen-specific responses, we found significant increases in the antigen-responsive CD4, CD8, and V gamma 9 T cells, shown here as total numbers on a log scale. Of cells expressing any of these cytokines shown on the axis, interferon gamma, IL-2, TNF, or IL-17. And those were measured for 3 months post-vaccination. So by 12 weeks after vaccination, what we're left with is about a log greater number of CD4 T cells compared to CD8s, and about a log greater CD8s compared to our V gamma 9 positive cells. Once again, 5 months after vaccination, these animals began receiving antibody infusions to deplete their T cells, and those infusions were continued throughout the challenge phase. And throughout the challenge phase, we monitored the extent of depletion in PBMCs, and then looked at the lung tissue at the time of necropsy. This slide shows the frequency of CD4 T cells in animals that received anti-CD4 depleting antibody in red, before depletion and after depletion compared to control animals, or the percent of CD8 T cells in blue in animals that received anti-CD8 alpha. We saw extensive and sustained depletion of both these T cell subsets in animals that received those antibodies. Moving to the site of infection, we looked in the lung tissue at the time of necropsy and found that CD4 and CD8 T cells were about 90% depleted in those animals compared to control depleted animals. However, I caution you that some residual T cells may remain, especially in the context of vaccination, since I just showed you that IV BCG elicits this large number of T cells into the bowel and into the lung tissue. So if we express these same data rather than a proportion, but as total numbers of cells in the tissue, you can see that 90% depletion still leaves about 5 times 10 to the 4 CD4 T cells per gram and fewer CD8 T cells. But these T cells could be antigen specific and could contribute to protection. So can IV BCG still protect animals against TB when they're depleted of CD4 or CD8 alpha cells? And that outcome is shown here. On this graph, I'm showing you lung CFUs at necropsy. And in this experiment, we saw 3 out of 12 animals that were IV BCG vaccinated, but not depleted, conferring sterile protection, with about 6 additional animals having a greater than 3 log reduction compared to our unvaccinated infected control animals. Animals that received IV BCG but were depleted of CD4 T cells, in contrast, had a significantly higher number of CFUs compared to these controls, and some of these animals had higher CFUs than even our unvaccinated control animals, indicating that CD4 T cells are in fact required for mediating protection after this vaccine. In our CD alpha depleted animals, we had a sort of split phenotype. Some animals had high CFUs and were not protected, whereas about a third of the group maintained protection after this depleting antibody, indicating either that CD8 alpha cells are critical in some animals and not others, or possibly reflecting an extent of depletion, differential depletion in these animals. Moving then to lymph node CFUs as an indicator of dissemination of TB from the lung to the lymph node, now we see that both anti-CD4 and anti-CD8 alpha treated animals had significantly greater CFUs in the lymph node compared to these controlled depleted animals that really are able to shut down the TB infection and prevent dissemination from the lung into the lymph node. So in summary, I've shown you that CD4 T cells are critical for IV BCG mediated protection. You saw the CFUs and dissemination, but this was also true when we looked at granuloma numbers, PET-CT inflammation, and pathology. CD8 alpha T cells are likely involved in control of MTB and dissemination to the lymph nodes in some animals, but not others. The caveats of this experiment are that residual CD4 and CD8 alpha cells can persist and may provide some protection in this model, and that the depletion with CD8 alpha targets unconventional CD8s as well as double positive CD4 and CD8 T cells. So we're currently halfway through a follow-up study in which we're trying to define the role of strictly conventional CD8 alpha beta T cells by comparing CD8 alpha and CD8 beta depletion in IV BCG vaccinated animals. But if these classical CD8 alpha beta T cells are important, that has important implications for vaccine design. For example, vaccine platforms such as viral vaccines, whole cell vaccines, or even mRNA might be more efficient at eliciting CD8 T cells than traditional platforms such as proteins and adjuvants. So with that, I'll stop and acknowledge our collaborators and the people who worked on this study, and also acknowledge that we had funding not only from the NIH Vaccine Research Center, but also from the Gates Foundation. Thank you.
Thanks so much, Dr. Darrah. That was really fascinating, and I certainly look forward to hearing about further developments in your research. Our next speaker is Dr. Alex Schmidt, and he serves as Head of Vaccine Development at the Gates Medical Research Institute. He joined Gates MRI in 2018 after 7 years with GlaxoSmithKline Vaccines, where he led vaccine development programs for dengue, respiratory syncytial virus, and influenza virus vaccines. Prior to GSK, Dr. Schmidt spent 10 years in the NIH intramural program, where he led a lab-based team and worked on translational and clinical development for viral vaccines. Dr. Schmidt received his MD from Freie University in Berlin. He trained in pediatrics at Charité and at the German Heart Center, and held a faculty position in pediatrics at Charité. I hand you over to Dr. Alex Schmidt. Thanks.
Thank you so much, Indira, for the kind introduction. So Willem and Lily asked me to talk a little bit about BCG revaccination, and that refers to vaccinating children or young adults in their second decade of life. You heard from Mark that BCG vaccination in the first week of life confers good protection for about a decade, but that there isn't much protection past the first 10 years. And I was asked to put this in the perspective of what's going on in TB vaccine development in general. So I'll give a little bit of an overview of what's happening and then focus in on BCG revaccination. I think most of us agree we would like to have a new vaccine that helps us accelerate the end of the TB epidemic. And the WHO defined a couple of preferred product characteristics for a vaccine that targets adolescents and adults as they drive the current epidemic in high burden countries. So they talk about vaccines for the prevention of disease and vaccines for the prevention of infection. And then there's a second set of target product profiles for an improved vaccine for neonates. And last but not least, there are product profiles for vaccines that prevent recurrence or increase the proportion of cure and accelerate treatment. I'll focus on adolescents and on prevention of infection, prevention of disease vaccines. So if we look at the national history of TB following exposure and infection, I think the picture that emerges is that we can't just talk about two boxes, latent versus active, but that there is a continuum, and that the bacillary burden here depicted in the red line and along the y-axis can go up and down, and it's basically your usual battle between the host immune system and the bug, whether you can contain the infection, clear the infection, or the infection takes over and you develop disease. And one huge challenge we have with TB in general is that our diagnostic tools are fairly limited, right? So we have the skin test, we have the interferon gamma release assay to look for an immune response to infection, but we can't detect the bug unless there is active disease. And that's a huge issue for vaccine development as well. And then we don't know how late in the progression of disease people are able to clear the infection or to contain infection, right? We've had recent data from using sensitive sputum PCR technologies that show that the walking well, having TB DNA in their sputum isn't that infrequent. And the question is, are they on their way to developing disease, or could they stay healthy for years and years to come? And then in that IGRA negative population, you see here in the blue bar at the bottom, again, that's a little bit of a mixed box. Of course, if you're uninfected, you're IGRA negative or skin test negative, but then there are people who are repeatedly exposed, the so-called resistors, who have antibody responses to MTB, but don't convert by IGRA. And the question is, if you clear the infection and you get rid of the bug, could you then turn to IGRA negative? Or is IGRA reversion from IGRA positive to IGRA negative an indication of a protective immune response? If we look at the pros and cons of these different endpoints for vaccine development and for prevention of infection, clearly the big benefit is that the incidence is high, right? You can have settings in South Africa where you see 5 and 8 and 10% IGRA conversion or infection per year in adolescents. So you can do with a small study and demonstrate efficacy. But as I said, we don't have a good bacteriological test to detect early infection. And only 1 out of 20 progresses to disease. So if you show a 50% reduction in infection rate, you can't say I will prevent 50% disease, right? Because you could possibly just protect the people who would have never progressed to disease. And so it'll be tough to convince health authorities that a 50% reduction in infection is likely to confer public health benefit. For prevention of disease, the biggest challenge is that the incidence rates are low. I mean, the burden is huge, right? It's the single biggest killer for an infectious disease globally, but the incidence rates are low. Lab confirmation can be done, but needs work, right? Sputum isn't the easiest matrix. You need high quality labs and you need them in settings where the burden is high. The incidence is highly heterogeneous, right? Think of South Africa, you can drive 10 minutes and the incidence drops tenfold. So you need to pick your sites for a trial carefully. But on the positive side, if you demonstrate prevention of disease, then I think health authorities will be easily convinced. And then prevention of recurrence, I won't talk to that much, but the incidence rates are higher, much higher than for POD. But the other challenges, differentiating recurrence from reinfection, impact of treatment regimens, and altered lung architecture on efficacy. So as Trisha mentioned, we really desperately need correlates of risk and correlates of protection to accelerate vaccine development, because we can't take every candidate into a phase 3 trial. There simply isn't enough money and resources to do that. So if you look at this clinical development pipeline here, it looks fairly rich and full, but in fact, we wouldn't be driving the Model T if it was a full pipeline. And that goes back to us not understanding mechanisms of protection well. I'll talk a little bit about the candidates here in the red boxes. In phase 3, we have a large trial in India sponsored by the Indian Council of Medical Research. It's a 3-arm trial comparing a recombinant BCG that is designed to be more immunogenic to an inactivated whole cell mycobacterial vaccine. For 4,000 participants per arm for a total of 12,000, and that's done in household contacts. And then you've heard last week on the left here in phase 2A, MTB-Vac, a live attenuated vaccine, will move into phase 3 for neonates and potentially for adolescents and adults as well. The 2 vaccines we're working on are here in the middle for phase 2B, and that's BCG Revax, and then the protein plus adjuvant program, M72ASL1. So I'll talk to those 2 a little bit. In 2018, this study was published, run by AHRI, a 3-arm study comparing BCG revaccination of IGRA-negative adolescents to a protein plus adjuvant vaccine to placebo. And while the study failed on the primary endpoint, IGRA conversion, there's significant protection from sustained IGRA conversion in the BCG arm. And sustained conversion was defined as initial conversion and then staying positive 3 and 6 months later. And now the question is, what does that mean, preventing sustained IGRA conversion, right? It implies, if you look here on the top right in panel D, the inverse of sustained conversion is early reversion, right? So you go from IGRA negative to IGRA positive and back to IGRA negative within 3 or 6 months. And that rate of reversion was doubled in the BCG arm. So I hope the hypothesis, if you want to be positive, is these people cleared the bug and therefore BCG revaccination conferred protection. And some of the more recent models take this self-clearance into consideration when they think through who's at risk of progressing to TB. But I think that's been a shift in the field to say not all TST positives or IGRA positives are at the same risk of progression to disease. The risk is highest in the first year or 2 after infection, and then it decreases considerably. So we set up a study together with Mark Hatherill and many others to basically demonstrate or test that observation from the previous BCG Revax study in a larger confirmatory trial. We're enrolling 1,800 adolescents, 10 to 18 years of age. They're randomized 1-to-1 to receive saline or placebo. We have a 10-week washout period to make sure nobody got infected just before vaccination. And then the kids are seen every 6 months for 4 years or for 4.5 years. If they convert, they have an initial conversion from IGRA negative to IGRA positive, we bring them back in 4 and 12 weeks later to do immunology work to understand what happens in that period of recent conversion and reversion, because that's the critical period we feel for control of TB infection. So last week the study was fully enrolled. You see here the blue randomized subject, 1,800 were enrolled, about 3,800 were screened. And the most common cause for exclusion was being IGRA positive, and that's at age 14 in poor poor communities in South Africa, 50% actually have TB infection, which is quite incredible. So the primary objective here is to demonstrate the efficacy of BCG revaccination for the prevention of sustained infection, as read by the AHRI test, and we'll trigger the efficacy analysis once we have 118 cases of sustained conversion. And then we'll look, of course, at safety and reactor and evaluate the durability of efficacy. And then a huge part of this study is to try and understand correlates of risks and correlates of protection. And the question really is, if you look at this schematic, moving kids from panel B to panel A doesn't help us with prevention of disease, because those are kids who would have never developed a disease. Only if we have enough, we move enough kids from panel C to panel A do we actually see prevention of disease. And so there's a large group of investigators working on this. You'll recognize many of the names here. For cellular immunity, Julie McElrath and Alex Schmidt, for instance, for innate, Elisa Nemes at Cape Town, people at Stanford and other groups. So really large collaboration to look at different types of immune response and to look for correlation with reversion from IGRA positive to IGRA negative. And at Gates MRI, this program is led by Nicole Fram. Then we have a similar program for a prevention of disease vaccine, and we're trying to link these two correlates efforts, right? So we'll look in the BCG revax from the previous study. We'll test the findings in the new study. We look at correlates in the M72 phase 2B study, and we'll try to test them in the phase 3 study. And if we can detect correlates of protection that are shared between POI and POD vaccine, then the question is that good enough to convince regulators that POI or POSI can be used as a surrogate, or other biomarkers can be used as a surrogate for large clinical endpoint trials? Here is a result from the phase 2B trial of M72 AS01. That was a trial in 3,500 IGRA positive adults, and 50% protection from disease was seen, which doesn't look so great compared to COVID, but looks great compared to everything else that's been done in TB vaccine development, because this is the first one since BCG where we have evidence of efficacy in man. So GSK, with the support of the Gates Foundation and AHRI, ran this program through phase 2B, and now Gates MRI took it on to get it into phase 3. And here you see the run-up. It involves a lot of manufacturing development, getting up to commercial scale, getting manufacturing robustness. We're running a phase 2 trial in people living with HIV, so that we have the safety data to start the phase 3 trial, including people living with HIV. We're starting a large EPI study to look at IGRA positivity by age at site level, and then select the sites with the highest force of infection. That's a study in 8,000 participants at 50 sites in 12 to 15 countries. That should kick off late this year. And then in 2023, we feel we're ready, and the bottleneck here is the technical, the CMC development. In 23, we'll start the large phase 3 efficacy trial. Here you see the initial proposal for the phase 3 trial design. We think this needs to be a large trial with up to 20,000 participants. Our base case is 18,000. We want 12,000 IGRA positives in that trial, so we can repeat the phase 2B results and demonstrate vaccine efficacy for POD. We also want to include about 6,000 IGRA negatives, so we can look whether the vaccine has POI efficacy. And then we want to include people living with HIV in adolescents and submit, and that's to be negotiated with regulators as early as acceptable. So how do we conclude on this? I think if you heard Mark's talk and Trish's talk, BCG revaccination clearly has potential for prevention of disease, but we just don't know it yet. And we critically rely on correlates work, both correlates of risk and correlates of protection, to make our case. The POD vaccines will take still years to read out on phase 3. And so if we can make that link between correlates for POSI and correlates for POD, then maybe we can convince policymakers or regulators that a second dose is beneficial. But I think there's a big question mark yet, or a gap in data, basically. But we do know we won't see 10 phase 3 trials in the next 10 years because they're too large and too expensive. Many vaccine candidates won't find the funding to go into phase 3. So there's hope and there's a role for BCG revaccination in that space. And thank you very much for your attention.
So thank you very much, Alex, for that phenomenal talk. In fact, thank you to all the speakers for magnificent talks. My name is Willem Hanekom. I'm at the Africa Health Research Institute, or AHRI, in KwaZulu-Natal in South Africa. And so now we have time for some discussion. If the speakers want to put their videos on, that would be great. I want to start off, Mark, with a question about safety of BCG. There are many, as you could have seen from Alex's talk, there are multiple BCG replacements that are currently being developed. Is BCG not safe then? Do we need replacements? Why are these vaccines being developed?
So a lot of the thinking around the safety of BCG, I suppose, has its origins in the early days of the HIV epidemic and the risks of giving BCG to infants who are perinatally infected. And I think that's probably primarily still the push for a safer infant TB vaccine. But of course, since that time, at least as far as infants go, we've seen much improved perinatal antiretroviral coverage. The incidence of HIV infection in infants has plummeted in most countries. So the safety concerns around these live vaccines for infants are certainly less. That said, BCG is a highly reactogenic vaccine. It leaves a nasty scar. So ideally, one would want a vaccine which is efficacious and not quite as reactogenic. If you offered me a vaccine that offered more durable protection, I'd take that above the current safety limitations.
Thank you, Mark. And I want to also upfront ask Trish a question, if I may. What is it about those CD4 cells that are protective?
Well, I guess we don't know. We were excited to see the data. Of course, there's a lot of people looking at different mechanisms of protection, and we try to look at those, at treated innate immunity and antibodies, and of course in our IV BCG model. But of course, we've been focused on T cells, and we have seen that IV BCG elicits high CD8 T cell responses as well, which we think is unique. But we were really excited to see the data, such definitive data in the depletion study, that CD4 T cells were in fact required for protection. And so, I don't, whether or not it's classical CD4 activation of APCs to kill those intracellular bacteria or not. We might be able to see something in, if we look at transcriptional data before and after our T cell depletion, we might be able to see a signature that's lost in those animals. But but I don't know if we know for sure. Also, I think whether or not we're going to know in the lung or the blood is, if we'll be able to see that signature in the blood is another question, or whether we'll have to look for things always directly at the site of challenge. So I guess some follow-up experiments that we're doing right now would be to really try to understand the early immune events after challenge, either by looking by microscopy by single cell analysis or possibly even a spatial seq approach to try to see if we can visualize or have a better understanding of what's happening in the lung and maybe what's really being recruited right around those infected APCs in the lung really early after challenge. So maybe we'll have some more data soon.
Thank you very much. I just want to alert people that 17 questions have already been answered, so I may not go back to them, but there are still some. So look in the chat box for the answers of these questions. But there is the inevitable question, Alex, has come up, that's been discussed over and over. Why are we doing so poorly, whereas COVID-19 vaccines could be developed in a year and they're all being rolled out? Millions, tens of millions of them, hundreds of millions. So what are we doing wrong? What are the barriers?
I can start, and I'd love to hear other panelists and you, Willem, on this as well. But I think one huge challenge is our limited understanding of the biology of MTB, right? For SARS-CoV-2, without having seen any data, if you ask 10 virologists what's the key antigen for protective responses, everybody would have said it's S. It's a surface protein and it's neutralizing antibodies. If you ask 10 microbacteriologists what's the single most important protein or lipid to use in a vaccine, I don't think you get this consensus. So it's a very complex bug. It's a disease that doesn't work itself out in the bloodstream or in the epithelial surface, right? It's deep in the tissue, and so understanding protective responses is really challenging. That's the biology part, and then of course all the rest that what people mention: lack of funding, it being a disease of the poor, there not being return on investment, and all the rest of it.
It's also a quick model, right?
Right.
It's also an easier model to test. So it's a quick pathogen. You can do challenge experiments and get an answer in 7 to 14 days. You can easily monitor infection by looking at viral loads, and so it's, I think, experimentally also easy to or faster to address.
Mark, do you want to comment?
Yeah, I'd love to. So when I look at the response to the global TB epidemic and compare it to SARS-CoV-2, the knee-jerk reaction coming from the eternally poverty-stricken TB vaccine community will always be, well, the issue is funding, and if we had $4 billion, then we could do much better. But that's I think simplifying the issues tremendously. I think if more funding were available, it would certainly allow many more candidates to enter the clinical pipeline and to be tested at higher risk, because that's a major limitation to the flow of candidates through to phase 3, as I think Alex mentioned. But the reality is that if we had a what looked like a very promising mRNA vaccine candidate, and we sent 20,000 doses to Gates MRI tomorrow, we would still probably need 5 or 6 or more years to get efficacy data in the absence of correlates of protection, because TB is slow, and we're never going to get data out of TB vaccine trials in this, at anything like the rate as we got them out of COVID-19 vaccine trials.
I mean, the difference in epidemiology, right, is also critical. The heterogeneity of incidence rates, and the fact that COVID trials could be rolled out across the globe, high resource, low resource, any region. It was a virgin soil outbreak with high incidence rates. And here for TB, while the absolute burden is mind-boggling, finding the high incidence sites isn't trivial.
And it's not just funding, because look at HIV, that's gotten more funding than anything, and you still don't have a successful vaccine after all this time. So it's a lot to do with the pathogen, I think, and that COVID wasn't, I mean, as devastating as it wasn't a tough nut to crack in terms of protection. Everything works, right?
I want to push back against the three of you and say that funding is a very, very big part of this. I think more than $110 billion has been spent on COVID vaccines. less than 1 billion, much less than 1 billion on TB vaccines. In South Africa, the amount of people that die from TB every day is about this, if you, on average, it's about the same as has died from COVID over the period that we've had COVID. And so we don't have the advocacy to mobilize to get that funding in place. I think that, and obviously the reasons are those that Alex mentioned about a disease that is socioeconomically dispossessed and with a poor voice. For example, if we don't know which protective antigens to use in TB vaccines, but if we had unlimited funding, we would have by now had multiple mRNA vaccines and have been testing these in animal models, I think. So I do think funding is a very big part of that, if I may push back a little bit, and advocacy for that matter. I want to get onto another question that's already been answered, but this comes up all the time when one talks about IV BCG. Should we not do a human trial to look at IV BCG, or at least do an experimental medicine study with IV BCG to look at protective mechanisms in the human? I don't know if, Trish, you want to comment, or anyone else?
Sure, we've talked about it a lot, and of course it's something that people always ask. Safety is a primary concern. We think about it all the time, and obviously people are concerned about the safety of giving a live bacterial vaccine intravenously. What I can say is, I guess from a scientific perspective, we like to say, if we have something that works this well, is there a way we can think about making it safer? But and that would be sort of using, do we use a more attenuated strain? Do we use a lower dose that we now know might protect just as well? Or even a heat-killed or oxytroph that might not replicate at all. I can say that there's some data that Chuck Skanga from the University of Pittsburgh has generated, which has been really exciting, that showed that IV BCG worked in SIV infected macaques, and that speaks to its safety a little bit, although he did treat with antibiotics after 2 months, I believe, to be sure those animals didn't have trouble with disseminated TB. So I think the feasibility is there, and the safety is something that we might be able to address. But there's a lot of hesitation, and we've gone to the FDA here in the US with some initial inquiries about whether or not what they would require to move forward with a trial, and they really wanted some intense monitoring to make sure that there weren't any long-term effects in the spleen. I know that VPM in Germany is also considering doing a human trial. So I think it would be amazing to be able to know what the immunogenicity and protection will be in people, but I also think that everyone is a little bit concerned with safety. I think we've given it to about 150 monkeys right now at that high dose, and have never had a death or a severe adverse effect that we, that in terms of monitoring those animals. So that's encouraging.
Thank you. Can I ask the next, multiple questions have been asked about the differences between blood and lung and BAL responses, or looking at the lung itself. And these may have huge implications for the ongoing correlate studies. Of course, a correlate is a correlate, and if it's validated, it is a correlate, and it becomes a surrogate of protection. But in terms of your data, Trish, that suggests that that one really needs to look at the lung if you want to know what's going on. What are the, and any of the panelists may answer this, what are the implications for monitoring for correlates of risk, perhaps in clinical trials? I think Lele also suggested that she would like to answer this. Lele Rangaka is one of the panelists here. Do you want to answer that, Lele?
No. I actually was suggesting that we get Tom to ask the question, but you have already asked.
Oh, okay. All right. So then it's over to the panelists to tell us what they think.
Just quickly, because I want the correlates people to talk more about this than me, but I felt a little bad showing that blood slide, but we just ran that data, and I wanted to include a little bit of it because it's new, but that's honestly a single single immune parameter out of the so many, the 1,000 things we could look at in terms of blood. And so we hope that we'll be able to find something else. But whether or not the signal is going to be more robust in the lung, I think is possible. Although I have to say we've had trouble discriminating in this dose study. Sometimes we see the signatures is really driven by the dose and not necessarily the protection. So it's been hard to disentangle the signatures that we see just with higher dose in animals that are protected versus not.
Yeah, I would say, since Tom asked the question, Tom is aware, but the study, the nested study in the current BCG revax study, Elisa Nemes, I believe, is the PI, and with collaborators at Stellenbosch to do BAL following initial conversion and maybe reversion. So looking at mucosal responses in the lung shortly after infection, I think is critical to further understanding of what's happening, right? 19 out of 20 do okay with the infection, and we need to understand what's happening in the lung early on. I think there's a bifurcation there probably.
Yeah, so I'm from the clinical trialist perspective. Should we do studies to understand the mucosal responses in the lung? Absolutely. Can we do so at scale? Absolutely not. But we can do small experimental medicine studies to inform our the development of whether it's BCG or other candidates.
Thank you very much for that. Then there is Alex. There are two questions that are about the development issues. The one is, would POI lead to POD? Okay, I think you addressed it a little bit, but perhaps we can get a little more input into that. And then a question about whether M72IP would have impact on development.
Yeah, on POI to POD, well, not sure if, let me ask if I get the question correctly. Is the question, should we use something other than disease as an endpoint for a phase 3 efficacy trial? Is the concern that the walking well who are PCR positive in sputum contribute to the epidemic and to spread? And I think that's an open question. It's hard to run a phase 3 trial without a disease endpoint, because you have, if you screened people on study, say once a year for sputum PCR, without suspected TB as a trigger, some people might like that, but it's really difficult how to use that data for a license application. I think you need to show, absent accepted correlates, you need to show a reduction in disease. And I think this area of PCR positive sputum without disease isn't quite well enough understood at the moment.
I think, Alex, if I may come back, I think the question is also, isn't it a given that if POI, prevention of infection, is shown that you're going to have a reduction in prevention of disease? Why do we have to even go ahead and do go all the way to prevention of disease? You've had lots of experience in talking to regulators and to even authorities within different countries about this. So
yeah, but it's really it's the lack of expressiveness, if you will, right? And because only 5% progress to disease. Now, if you reduce infections by 50%, you may still have 5% progressing to disease, right? Because we don't understand who it is that progresses. If you had 100% POI, you could conclude you had POD, but shy of 95% POI efficacy, you can't conclude that you will see POD, right? It's a chicken and egg problem. Aren't there, right? I mean, there is a genetic component of who progresses and who doesn't. So the people who are protected might be the ones who wouldn't have progressed in the first place.
Yeah, I agree with Alex. and anything short of 90 to 95% protection in a prevention of infection trial is always going to raise the doubts around whether that, to what extent are that 5 to 10% of individuals who progress to TB, to what extent are they covered?
That's a high bar, Mark. So can I ask the next question? There's a question about controlled human infection models, given that that the NHPs are astronomically expensive and the sample sizes are often right at the edge. What's happening in this field? Alex, do you want to perhaps answer that?
I think
Trish?
Mark might know more about this, or you, Willem, than me. I'm aware of the BCG challenge models. If you think about pulmonary challenge, I am not aware of a model that is valid. And if it's a BCG skin challenge model, then people will raise questions around extrapolation to pulmonary TB. But I'd love to hear you, Willem and Mark.
Yeah, I'm aware that models are in development. I think the issue is that we don't have the ideal model that can be implemented.
Yeah, I think my perspective is that it's also not been an easy route to develop the actual bugs that can be used safely in humans. There are various demands on that bug that would have to be used in the challenge if it's not BCG, such as the studies that Helen McShane are doing. It's a difficult path, but I think there's a lot of effort that's going on in this field exactly for that reason. Further question, I don't know who wants to answer that, perhaps Mark. In the UK, it appears that the majority of the increasing cases of TB relate to reactivation in adults rather than primary infection. Is this true elsewhere, and may it explain the lack of efficacy of BCG in adults?
Yeah, there are a whole lot of moving parts there. The different countries, the epidemic in China and in certain parts of Europe is very different from what we're seeing in places like South Africa, where a lot of the TB is arising from recent infection. I don't think it's a matter, an issue of age. It's more the issue that, as we know, the highest period of risk for progression from infection to disease is in the first 1 to 2 years after infection, and thereafter there is some risk, but it's and that's the issue with identifying IGRA positive or TST positive people for inclusion in trials, right? So we're including some people who have recently been infected and a whole lot of people who've been infected potentially a long time ago. So yeah, there are a lot of moving parts to that question.
And perhaps I can then follow up with, and I know there's very good modeling data available to answer this question. What do the panelists feel is the best protective disease efficacy that a TB vaccine can achieve in practice? Is getting to 80% possible? And perhaps I'd like to link to that. What does the modeling show in what would already have a huge impact in the TB world? Perhaps Alex, do you want to start with that?
Yeah, sure, happy to start. Is 80% possible? I think we have to aim high. Is it feasible with the current candidates in late stage? It'd be optimistic, right? If you look at the efficacy of M72 AS01 with different primary endpoint definition, it ranges from 50% for the primary, and then in the sensitivity analysis, it goes up to 68% if you're more stringent with regards to lab confirmation. So 68 is also already a lot better than 50. If you look at the modeling data, if you see 30%, 40%, you already have significant public health impact. right? So we shouldn't think of the vaccine as a failure if we get to 50%. We'll see public health impact if we can get implementation and rollout.
Yeah, if I can add to that, I think implementation is a huge issue that we tend not to think about too much, and until it's somewhat late in the day. We tend to take the number from the efficacy trial and work with that, but the reality is, depending on the schedule and how easy it is to find and engage the target population, the real effects on in communities and in countries may be very different from what we initially projected. And you can see it now in the COVID-19 vaccine rollouts. A single dose vaccine is a different issue from a 2-dose vaccine, and there are issues around vaccine hesitancy and even in settings where disease prevalence and incidence is high, and the risks of acquiring that disease are also high. So there are a lot of factors to consider in terms of the practical implementation.
We thought about that in the monkey model as well. Sorry, Willem. Just about what level of protection would be sort of a go decision for a human trial. So we were excited, of course, with the IV BCG data that it was so dramatic. But do you need sterile protection, or is even with our high dose intradermal, we did better than the low dose intradermal? We were able to further reduce CFUs. So can something with a couple log reduction in this really susceptible rhesus model have a meaningful impact if you move to humans? And then that gets into bigger conversations, I think, about using this really stringent rhesus model compared to testing vaccines in something like a cyno model that might more likely reflect humans, where you might see they might be easier to protect, but then you're forced to use like larger animal numbers and the experiments get more expensive. So it's a tough question to know in monkeys too, what protection should you shoot for? And just one other thing there is that we often see this kind of all or none protection in the rhesus model. So it's either really great or the animals do really terrible. And so there isn't a lot of gradation in that model to understand what might be partial protection.
So I'm going to ask the last question myself to follow up on this, Trish, before I hand over to Lele. Why do we see that all or nothing effect in all your studies? What's going on there?
Yeah, I wish Joanne were here because she thinks about this a lot, but it really does seem to be, we have a couple animals that sometimes have low CFUs, but otherwise they tend to do really bad or really good. We also are only following them for 3 months. So we don't even know whether animals that have just say 50 CFUs at necropsy are going to go on to protect or go on, to do really badly. So we're not sure whether you if you don't have that sterile protection in the very beginning, will you still get control? Although Lewis's data would suggest that that's possible with the CMV vaccine, right? And so it might be just the rhesus model. Yeah, I don't know if it's just the rhesus model. We'd really like to do these experiments in cynos and see if we see the same effect.
Thank you very much. Over to you, Lele.
Thank you. So let me just speed it up. Okay. So now it's been an incredible hour and a half, and thank you so much for that excellent discussion. So we've journeyed, from the past and well into the future, looking at what novel TB vaccine development looks like. And I just want to thank our speakers, Mark, Trish, and Alex. Thank you so much for taking us on that journey. And really thank Hanif, Indira, excellent moderation, and definitely with Willem as well. So from all of us at UCL-TB and also AHRI, thank you. And we hope that we will not let the lack of commitment prevent us from advocating for more work, more excellent science, so that we can actually get the TB burden under control. So thank you so much. Have a good day.
Thanks for having us.
Thank you.